Already discussed in our previous class about plant breeding steps and also learn about plant breeding importance in the agriculture. Okay, with help of the plant breeding process, new varieties of high yielding crops and disease resistance plants and pest resistance plants and also improve food quality. Food quality crops are introduced in the agriculture field with help of the plant breeding techniques. Now in this class, I want to go to discuss briefly about the last previous video concept and also added some easy learning video clips. Okay, let us discuss about the plant breeding process. Okay, first of all, India is an agriculture country because so many people are depending on agriculture as employment. Okay, in our country, 33% of gross domestic product is obtained in the agriculture field, and also approximately 62% of the population in our India mainly depending on agriculture as employees. Understand? So we have limited land. Okay, in ancient time we have limited farmland, but population is also gradually increase, but the farmland is not increase. So we introduced high yielding per unit area crops. This means increase yield per unit area to introduce new varieties of crops. In that situation, we are facing the food supplement throughout the India. Because of this reason, in 1962, 2000 years, in this period, our agriculture yielding is gradually and dramatically increase with the help of the plant breeding process. In this plant breeding process, a new varieties of crops are introduced in the agriculture. So in this period, a dramatic increase crop yield and dramatic increase resistance to the plant, dramatic increase stress quantity of the food materials. With help of the Norman Borlaug, M.S. Swaminathan, there are fathers of Green Revolution. So this increasement, that's a dramatic increase crop yield in the agriculture process. This is phenomenon is known as Green Revolution. Next one. With help of the plant breeding process, so many different types of high yielding crops are introduced in the agriculture field in our country also. Next one, first one, in the wheat, Godum, Sonalika and Kalyan Sona. These are highly resistant and also give high yielding Mexican wheat varieties. These Kalyan Sona and Sonalika wheat Mexican varieties are introduced in our India by the M.S. Swaminathan. Because of this reason, our wheat yielding is increased 11 million tons to 75 million tons. In rice crops, IR8 and Tavanchung 90 -1. These are wild varieties and also semi-drop varieties like Jaya and Ratna gives high yielding. This type of semi-drop rice varieties Jaya and Ratna is introduced in our country. Because of this reason, our product is increased 35 million tons to 889.5 million tons. In sugarcane, in sugarcane. Sakaram barberry, it is a North India crop, this one. Sakaram officinarum, it is a South India sugarcane. This is North India, so Sakaram barberry is a low sugar content and also low yielding. This one Sakaram officinarum, it is give high yielding and also stem analogy. Thicker stem is also present. So this Sakaram barberry and Sakaram officinarum both are crossed to form a new variety of crop. This crop is successfully grown in North India and also consists of thicker stems and have high sugar content. High sugar content. And mills, chirudhanyalu, mills, hybrid maize are also introduced with help of the plant breeding. Jowar, bajra, hybrid maize. This type of high yield varieties of mills introduced in the agriculture field. So these jowar, bajra and hybrid maize varieties are consist resistance and also water stress. Plant breeding process is helpful for disease resistance plant production. Okay, this plant breeding technique is utilized for the plants have disease resistance. Suppose different types of pathogens are causes different types of diseases. So like brown rust of wheat. It is caused by the Fuchsinia tritica fungal organism. Another one, red sugarcane. It is also caused by Coleotrichum falcatum fungal organism. Late blight of potato. This disease is caused by Phytophthora infestans fungal organism. So this type of brown rust wheat and red rot of sugarcane, late blight of potato, these diseases mainly caused by the fungal organism like Fuchsinia tritica, Coleotrichum falcatum, Phytophthora infestans respectively. Some bacterial organisms are also causes disease like black rot of crucifix. This black rot of crucifix is caused by the Xanthomonas comprestis bacteria. Because of this reason, yielding is also completely decreased in crucifix. Viral organisms. Viruses are also causes different types of diseases in the crops like tobacco mosaic. The tobacco mosaic disease is mainly caused by tobacco mosaic virus. And turnip mosaic. This turnip mosaic virus disease is also caused by turnip mosaic virus. In this way, different types of viruses, bacteria and fungal organisms causes different types of disease. So this disease is successfully resistant to the plant breeding technically modifying organisms. Conventional breeding and mutational breedings, these are helpful for the formation of disease resistance plants. Okay, suppose some chemicals and gamma radiation. Some chemicals and gamma radiations are utilized to create mutations. That means the DNA base pairs are changed. with help of these changing, plant exhibit some advanced characters compared to their parental organisms, the parental plants. Okay, suppose bhindi, it is a lady finger. It is mainly suffering with the yellow mosaic virus. This one is the lady finger. Okay, bhindi, yellow mosaic virus. This one is a wild species. These wild species are change into some chemicals and gamma radiation and change into a new variety of bhindi. This one is Prabhani Kranti. This Prabhani Kranti bhindi plant is highly resistant to yellow mosaic virus. The plant breeding process is also helpful for resistance to insects and pests. Insect pests. Okay. Suppose different types of several insects and pests are also causes different types of disease and also damage the crop production and dramatically decreased with help of these insect and pests. So these resistance, plant breeding resistance plants are also resistant to insect and pest. Suppose brassica, suppose rapicide and mustard. These brassica highly resistance. Understand? These brassica highly resistant to new varieties were formed with help of the plant breeding process. It is called Pusa Gaurav. This Pusa Gaurav is highly resistant to some aphids. Another one, flat beans. The new variety crops are produced or created with help of the plant breeding process. That one is Pusa Sem2 and Pusa Sem3. These are highly resistant to jesseeds, aphids, and also fruit borer. And another one, Wakra. This one is a lady finger. The new variety was Pusa Sauni and Pusa A4. These are highly resistant to soot and fruit borer. Breeding process is also helpful for food quality improvement. The food quality is also improvement with help of the plant breeding process. Okay, more than 840 billions of people in the world do not have adequate food meet their daily food and nutritional requirements. The nutritional requirements is suffering by these people. Okay, so this phenomenon is known as a hidden hungry. Hidden hungry means nothing but in our daily eating food is not sufficient proteins, vitamins, and amino acids, etc. So because of the lack of these high quality of or sufficient protein and sufficient mineral and sufficient micronutrients, sufficient fatty acids, we are suffering with the hidden hungry. So because to improve the quality or quantity or micronutrients, macronutrients, proteins and fatty acids, etc. are increased in the modified crop fields. ఒక క్రాప్లో మనకు కావాల్సిన ఫుడ్ మెటీరియల్ అయినప్పుడు అందులో క్వాలిటీని క్వాంటిటీని ఇంక్రీజ్ చేసే ప్రయత్నం చేస్తాం. దస్ టైప్ ఆఫ్ ఫెనామినా ఇస్ నోన్ యాజ్ బయోఫోర్టిఫికేషన్. This biofortification which ends breeding crops with higher levels of vitamins, higher levels of minerals, higher levels of proteins and higher than fats. Okay, this phenomenon is known as biofortification. And this plant breeding process, nutritional quality undertaken with the objectives of improved. Suppose protein content is also increased and oil content and also quality is also increased in this crops, and vitamin content is also increased in this field, and micronutrients and mineral content is also increased in the biofortification modified in crops. Understand? In 2000 year, different types of maize varieties also increased. Different types of maize varieties. Some examples of these maize varieties, you are increase the lysine amino acid and also tryptophan amino acid. Lysine amino acid and tryptophan amino acid. In Atlas 66 wheat variety, in this variety, the protein quantity, the protein quality is also increased in this crop. Next one, the golden rice. You know already, golden rice, it consists high vitamin A. Okay, we are taking in these foods like golden rice, we are not suffering with blindness. We are taking Atlas 66 wheat product, we are not suffering with any protein deficiency. We are taking maize, hybrid maize variety food, we are not suffering with lysine and tryptophan amino acid deficiencies diseases. Breeding process, Indian Agriculture Research Institute. Where is it? New Delhi. This Indian Agriculture Research Institute is also introduced, released several vegetables, crops that are rich contain vitamins, minerals, etc. etc. Suppose vitamin A, enriched carrot, spinach, pumpkin, pumpkins are also released. Vitamin C enriched bitter gourd, bathua, mustard, tomato is also released. And also iron and calcium enriched spinach and bathua is also released by the Indian Agriculture Research Institute. Indian Agriculture Research Institute. Next one, the protein enriched beans. Protein enriched beans are also released by the Indian Agriculture Research Institute, like Bod, Lab, French and garden pea. These are the different types of quantity and quality foods are also improved with help of the plant breeding process and also increased the yielding in our agriculture field in our India. So the India is not imported wheat and rice nowadays. The India is increased high quality food and increased high yielding and also help this quality and quantity improvement of the yield. We are export the materials, export the rice, wheat, different types of crops from India. This one is the ability of India to improvement of the agriculture crops with help of plant breeding process.
Plant breeding for disease resistance. One of the major problems for agriculture are pathogens and disease-causing agents. Almost 5 to 15% loss in the crop yield is due to plant diseases caused by a wide range of fungal, bacterial, and viral pathogens. An epidemic form of a pathogen can even destroy the entire crop. Let us look at few pathogens that cause diseases in food crops. First, let us discuss about some fungal diseases. Fungi are the primary cause of disease in crops. A fungus called Puccinia triticina causes a disease called brown rust in cereal crops like wheat, barley, rye, etc. The disease affects stems, leaves, and grains of the plant, leading to tremendous loss in the crop yield. Another fungus called Glomerella tucumanensis causes a disease called red rot in sugarcane. This disease mainly affects the stem and considerably reduces the quantity and quality of juice. Another fungus called Phytophthora infestans causes a disease in potato called late rot. It is the most deadly disease of potato crop. Let us now discuss few bacterial diseases. A bacterium called Xanthomonas campestri causes a disease called black rot in crucifers. Crucifers are leafy vegetables like cabbages, garden cress, broccoli, etc. The disease looks like a yellow-brown patch on the leaves, which slowly spreads to other parts like veins, roots, and stems as time passes. Viruses also cause diseases in plants. Let us now discuss about some viral diseases in plants. A virus called tobacco mosaic virus causes a disease called tobacco mosaic in tobacco and other plants. The disease forms white and black or sometimes yellow and green patterns on the leaves, which looks like a mosaic. This causes malfunction of leaves and other growing parts. Another virus called turnip mosaic virus causes a disease called turnip mosaic in cruciferous plants. This disease forms pale-colored ulcers on the leaves, which stunts the growth of the plant. There are a number of factors that are responsible for the development of diseases in plants. Some of these factors are host genotype, pathogen genotype, and the environment. Some of the pathogen genotype is even capable of developing newer and newer strains every time. Therefore, it is very important to incorporate the trait for disease resistance in breeding programs. Resistance of the host plant is its genetic ability to prevent the pathogens causing the disease. If the plants have disease resistance, it would reduce the use of chemicals like fungicides, bactericides, which can cause toxicity, thereby increasing the farmer's profits. There are different breeding methods for incorporating disease resistance. Let us first discuss the conventional method. First, we screen the germplasm for the source of resistance. Then we carry out artificial hybridization of the high-yielding crop varieties with these disease-resistant varieties. The progenies are selected and evaluated for high yield, disease resistance, quality, and other features. At last, the progeny are tested and released as new varieties. Here are some of the disease-resistant crop varieties bred by hybridization and selection. Himgiri is a variety of wheat bred to show resistance to fungal diseases like leaf and stripe rust and also hill bunt. Pusa Swarnam, also called as Karan Rai, is a variety of brassica bred to show resistance to white rust disease. Pusa Shubhra is a variety of cauliflower bred to show resistance to black rot and curl blight black rot. Pusa Komal is a variety of cowpea bred to show resistance to bacterial blight. Pusa Sadabahar is a variety of chili bred to show resistance to chili mosaic virus, tobacco mosaic virus, and leaf curl. Adding disease resistance by this method is subjected to many limitations. In this method, we can add disease resistance only if the disease resistance allele is present in the germplasm. Generally, there will be only limited number of disease-resistant genes in the germplasm. Due to this, the plant will show resistance to only few number of diseases. Genetic engineering. Due to the improvement in genetic engineering, we were able to overcome the limitations of conventional breeding. Genetic engineering allows us to incorporate resistance genes from unrelated species into the plant genome. For example, a resistance gene, RX01, from maize plant was incorporated into rice plant by this method. This gave the rice plant resistance to a bacterial disease called bacterial street disease caused by pathogen called Xanthomonas oryzae. Similarly, numerous other resistance genes can be incorporated into a single plant. Note, by this method, we could incorporate artificial genes as well as genes from microorganisms into plant genome. Mutation breeding. Another method of creating disease resistance is mutational breeding. Here, we use chemicals and gamma radiations to induce targeted mutations in the selected plants. This sometimes creates desired characters which are not found in the parent plant. Since these changes are heritable, the plants with desired characters are then selected and used as a source in breeding of new plants. This type of breeding is known as mutation breeding. For example, in mung bean, resistance to yellow mosaic virus and powdery mildew were induced by mutations. Plant breeding for developing resistance to insect pests. Apart from diseases, the other main challenge to agriculture is pests and insects. The pests and insects feed on the plant parts like leaves, stems, flowers, etc. This makes the plants easily susceptible to plant diseases. Some pests feed on the sap of the plants, causing wilting of plants. Sometimes, insects also act as vectors for disease-causing pathogens like viruses, etc. Overall, insect attack leads to heavy losses in crop yield and quality. Generally, in order to protect the crops from pests, farmers normally use pesticides and other toxic chemicals. This not only kills the pests, but also kills the useful organisms around the plant. Pesticides, also called mild toxicity in the plant, produce and lowering its quality. Moreover, these pests gradually develop resistance to the pesticide and continue to attack the plants. Some plants have naturally developed some morphological, biochemical, or physiological characters which prevent pests from attacking them. Now, let us have a look at the morphological methods. Wheat plants and cotton plants have hairy leaves that prevent jassids and leaf beetle from eating them. Solid stems in wheat are not preferred by the stem sawfly. Bollworms are not attracted to smooth-leaved and nectarless cotton varieties. The features of plants that make them undesirable to the insects for food, shelter, reproduction, etc. are called antizoonosis or non-preference. Now, let us have a look at the biochemical characters of plants. Certain plants like maize have high aspartic acid, low nitrogen and low sugar content that make them undesirable to the stem borers. All these traits act as sources of resistance genes for plant breeding. Some crop varieties developed through hybridization and selections for insect pest resistance are Pusa Gaurav, a variety of brassica that has resistance to aphids. Pusa Sem2, Pusa Sem3 are varieties of flat bean that have resistance to jassids, aphids and fruit borers. Pusa Savan and Pusa A4 varieties of okra, bhindi, have resistance to shoot and fruit borers. Improved food quality. Even though there has been a tremendous increase in the world food grain produce, still a large section of population is lacking proper nutrition and quality food. Out of 7 billion, more than 840 million people in the world do not have sufficient food to meet their daily nutritional requirements. Apart from that, it is estimated that about 3 billion people suffer from a condition called hidden hunger. Hidden hunger is when people are able to fill their stomach and they don't feel hungry anymore. But the food they ate had a lot of calories but lacked in essential micronutrients required for the development of the body. Hidden hunger happens when people consume more amount of staple foods like rice, wheat, jowar, etc. and completely neglect foods like fruits, legume, vegetables, meat, fish, etc. that are rich in minerals, vitamins, fiber, etc. To overcome this deficiency, breeders and biotechnologists have tried to increase the oils, proteins, vitamins, and mineral content in the commonly consumed food without reducing the productivity. This approach is called biofortification. Biofortification is the process by which the nutritional quality of food crops is improved through agronomic practices, conventional plant breeding, or modern biotechnology. Biofortification has led to the development of golden rice and super banana that is rich in beta carotene and alpha carotene to overcome vitamin A deficiency. Here are some of the examples of hybrids crops with improved nutritional quality. Lysine and tryptophan-rich 3 varieties of maize are Shakti, Ratan, and Protina. The high protein-rich old wheat variety, Atlas 66, has been transferred to currently cultivated varieties. Iron-fortified rice variety containing over 5 times iron and vitamin A has been developed recently. Vegetable crops rich in vitamins and minerals have been released by Indian Agricultural Research Institute, New Delhi. These are vitamin A enriched carrots, spinach, pumpkin. Vitamin C enriched bitter gourd, bathua, mustard, tomato. Iron and calcium enriched spinach and bathua, chenopodium, and protein enriched broad beans, lablab, French beans, and garden peas.
One is SCP, single cell protein. The single cell protein, conventional agriculture, production of cereals, pulses, vegetable and fruits, etc., may not be able to meet a demand of food at the rate of which humans and animals population is increase. Human and animal population is increase. So your increase, suppose our body is necessary for protein. So you are eating pulses, cereals, vegetable and some fruits, etc. But more proteins are available in meat, animal body. More proteins are available in the single cell organism. Suppose 3 to 10 kgs of grains oka animal తీసుకుంటే it produced 1 kg of meat. The grass తీసుకుంటే 1 kg of meat, 1 kg meat మాత్రమే ఫామ్ అవుతుంది. Okay, suppose 25% of human population mainly suffering with hunger and malnutrition. So this SCP is very essential nowadays because the human beings are mainly suffering from hunger and malnutrition. 25% of human population is mainly suffering with malnutrition and also hunger. Because of this reason, SCP are mainly essential for protein production. Okay, SCP is the alternative source of protein for animal and also human beings. మన బాడీకి సఫిషియంట్ ప్రోటీన్స్ కావాలంటే పల్సెస్ తీసుకుంటాం. అంతకన్నా ఎక్కువ ప్రోటీన్స్ కావాలంటే చికెన్, మటన్ ఆర్ బీఫ్ ఎక్సెట్రా తీసుకుంటాం. అందుకన్నా ఎక్కువ ప్రోటీన్స్ క్వాలిటీ కావాలంటే వీఆర్ డిపెండింగ్ ఆన్ SCP. SCP is the alternative source of proteins for animals and also human beings. So some algal organisms and also fungal organisms and bacteria are also used as SCP production, single cell protein production. Some of the algal organisms like Spirulina maxima, Chlorella, Pheronoides and Synendesmus, Synedesmus. This Synedesmus and Chlorella phyronoidina and Spirulina maxima. Spirulina, Chlorella, Synedesmus. These algal organisms are also used as for SSCP production, single cell production. And some fungal organisms are also utilized for the production of SSCP. What are they? Candidata utilis. It is commonly called Torula yeast. Torula yeast. The next one, Saccharomyces cerevisiae. It is commonly called Baker yeast. Baker yeast. Man bakery lo bread say the area because of the fermentation of the Saccharomyces cerevisiae. Next one, Chitominium celluliticum. Chitominium celluliticum fungal organisms. These fungal organisms are especially used for the SSCP production. Next one, some bacterial organisms are also utilized for SSCP production. What are their? Bravibacterium Chytoglutamimicum. Bravibacterium Chytoglutamimicum. This bacteria is also used as SSCP organism, and Methylophilus methylotrophus. Methylophilus and methylotrophus bacteria is also used as SSCP production. So these algae and fungi and some bacteria organisms are used as SSCP. Suppose Spirulina maxima. Spirulina maxima. Spirulina maxima can be grow easily on materials like waste, water from potato processing plants. This means containing these potato wastage process consists of starch. Okay. Next one, animal manure and even sewage to produce a large quantity and can be survived food which is rich protein in that Spirulina maxima. Spirulina maxima. Suppose a cow, 250 kgs cow produced 200 grams of proteins per day. ఒక రోజుకి 200 grams proteins మాత్రమే ఒక 250 kgs of cow produce చేసింది. అదే 250 grams of microorganisms. What type of microorganisms? Methylophilus methylotrophus. Methylophilus methylotrophus. These Methylophilus and methylotrophus microorganisms produced 25 tons of proteins. Not 200 grams of proteins. 25 tons of proteins. Okay? 250 kgs ఉన్న cow produce చేసేది 200 grams protein మాత్రమే. అదే 250 grams of microorganisms అయితే they produce 25 tons of proteins. Okay? Very large protein product is obtained from the microorganism. Because of this reason, SSCP are very popularized nowadays, and space travelers are also utilizing this product, SSCP product, in the form of powdering. Understand? The powdering. The space traveler వెళ్ళేటప్పుడు వాళ్ళు ఎటువంటి ఫుడ్ మెటీరియల్ తీసుకెళ్తారంటే SSCP. ప్రోటీన్స్ కావాల్సినటువంటి ఫుడ్ SSCP కాబట్టి SSCP తీసుకెళ్తారు. Okay? This one is the concept of the SSCP. Thank you.
The tremendous increase in world population has led to a shortage of food supplies. About 1/4 of world human population is suffering from hunger and malnutrition. The conventional agricultural produce of cereals, grains, vegetables, and fruits are not able to cater the demand of food and resources. Therefore, alternative methods of raising food items have to be evolved. One such method is single-cell protein. Single-cell proteins are the dried cells of protein-rich microorganism which are used as protein supplement in human foods or animal feeds. For example, the blue-green algae Spirulina and most common fungi mushroom. Spirulina is not only rich in proteins but also contains sufficient amounts of fats, carbohydrates, minerals, and vitamins. It is grown in starch-rich wastewater from potato processing plants, straw, molasses, animal manure, and sewage. This also reduces environmental pollution. The most common source of single-cell protein is single-celled yeasts like Candida utilis and filamentous fungi like Fusarium graminearum. A number of edible fungi like mushrooms are also consumed as food by people. Single-cell proteins are rich in protein and low in fat content, which is a desirable feature of human food. Almost 40 to 55% single-cell protein body mass is made up of proteins. Some bacteria like Methylophilus methylotrophis has 80% protein content. Tissue culture